Hello, everyone. I realized I joined before I was like messing with my lighting, and we will get started in just like 1 minute at the top of the hour. If you have any questions, please do type them into chat. There should be a chat box. I see people coming online right now. So again, welcome to the webinar where today's topic is going to be compensation in FlowJo version 10.10. And yes, Maria, the recording will be up in a couple of days on the FlowJo recorded webinars site, and I will point you to where that location is. So if you have to leave, if we go over the time doing some demos and discussing any questions that you ask, that will be available here in just a couple of days for on-demand watching. So it is the top of the hour. I think we'll get going. I'm going to share my screen and I'll start with a presentation with some slides. And I'm going to try and go really fast so that we can get to the live demos. I have a couple different data sets to show you. If I do mute myself and cough, it's because I've been sick. Everybody has over the holidays. And, my family has gotten a couple rounds of ick. So I'm still coughing and I apologize if that happens. I will try to limit that, but nothing much I can do. Let's go ahead and get started. I'm going to share my screen and we will go ahead and go into full view mode with this presentation on compensation tools in FlowJo version 10. Again, I see people coming online. Welcome to the conversation here. We're going to talk about compensation in FlowJo and go through all the tools and show you some of the bells and whistles available to, evaluate, how good to create good compensation and evaluate whether that's appropriate for your panel design or your instrument setup and such. So let's go ahead and get to it. Let me go into full view mode here. Usually, see it right here. There we go. Okay. Meeting is being recorded. It will be available online in a couple of days in the FlowJo website under recorded webinars, and I'll point you to that link at the end of this presentation. So my name is Timothy Crawford. I am the Senior Manager of Product Innovation at BD Life Sciences Informatics, formerly known as FlowJo. We're a wholly owned subsidiary of BD now, and I will be talking about compensation tools. As we go through, please feel free to type anything you want into chat. And I will be checking chat off and on so I can answer those questions. My basic outline here is I'm going to go through what compensation is and then talk about traditional compensation, where you gate positive and negative to define the populations that you use for each color to determine spillover coefficients and create the compensation matrix. And then talk about AutoSpill compensation, which essentially removes the need to gate positive and negative. It does linear regression to get the spillover coefficients and then does the calculation just like traditional compensation. Additional options, I'm going to talk to you a little bit about the spillover spreading and total spreading matrices that you can access once you've calculated compensation with your single color controls here in FlowJo. And to start off, we're just going to talk about, the basics of compensation. Compensation corrects for fluorescence spillover. And I'll explain a little bit more about what that means, fluorescence spillover, but you basically have a single channel where the fluorescence is going to be detected highest, and then you have lots of other open channels. Those are called secondary channels or secondary detectors, where the emission spectra of the fluorochrome in question spills into the other detectors and gets picked up. And so we have this example here, Alexa 700 spilling into the APCH7 detector. In the uncompensated state, you see the skewing of events. That is not coming from any signal from an APCH7 fluorochrome, but it's actually coming from the spillover of the Alexa 700 into the APCH7 channel. So when we apply our compensation matrix that we will calculate, we are going to correct for that spillover and bring this population of positive events down so that the positive and negative fractions of this single color compensated control have equal MFIs, or median fluorescence intensities, in the spillover channel, and that is a correctly compensated sample. So the basic need of compensation is there for every single multicolored panel, whether you do 2 colors or 30 colors. And each color needs its own single stain control. So each fluorescent probe, that could be an antibody conjugated to a fluorochrome, targeting a specific antigen, or it could be a dye that targets the cell membrane or the cell or DNA, right? Every fluorescent probe you use has to have a single color control so we can capture the emission of that color and then correct for the spillover across all the detectors that you have enabled on your instrument. This process results in removal of the signals that are contributed, as I said, from by non-primary fluorescent probes in a multicolor experiment. So into all the detectors that it's not supposed to be picked up in. And compensation is applied on a per event basis, right? So you apply a compensation matrix that shifts each event based on that matrix into a new position. And you should always be analyzing the compensated parameters in FlowJo. They are denoted by a comp prefix in front of the detector name. And then this label here, the antigen, is actually the stain reagent field. So you have the name of the parameter, the name of the stain that you can enter, and whether it's compensated or not is denoted by the presence or absence of the comp prefix. So spillover completely depends on the reagent and its emission spectra. So here I have a single PE size 7 emission spectra. It's being lit up by a yellow-green laser. And you can see it has this instrument, these boxes are denoting the different detectors or channels. So these are regions of light that are picked up by a filter. And each emission spectra is going to have a primary detector. That's where it has the peak emission. The most light is getting into that detector. And then a bunch of secondary detectors that it's spilling into. So everything else other than the primary peak area of the spectrum is considered a secondary detector, right? And what we're trying to do is calculate how much of the light coming from this emitted fluorescent probe or fluorescent molecule is captured in each secondary detector relative to the primary, which we set at 100%. So it's a fractional percentage of this emission is being picked up by each additional secondary detector. Now, as you grow your panels into more colors, the calculation becomes more complex. You have to get the spillover coefficients for each of these fluorescent probes or emission spectra of your dyes. And then you have to create a matrix. And this solution to solving how much fluorescence goes into each detector and removing that additional fluorescence into the secondaries requires a matrix math problem or a series of linear equations. So I'm not going to focus too much on the math. I'm going to give you a little bit of math, but it's not my specialty. I'm an immunologist. You're probably mostly life scientists. Let's let the computer do the math. We can just concentrate on the end results that we get out of performing these calculations. So I have a very simple 3-color experiment that I'm going to illustrate here to talk about how we actually calculate spillover coefficients and compensation itself, right? So the compensation matrix. In this example, I've got 3 different colors, 3 different fluorochromes attached to a cell, right? And we have 3 different detectors on this instrument. And my blue detector, right, or my blue fluorescence spectrum gets picked up highest in detector 1. So we call that 100%. And then some fractional percentage of that emission that is detected in D1 is also picked up in D2 and D3. So 12% in D2, 12% in 1% in detector 3. My green comes in highest in detector 2, so that's set at 100%. 15% is in D1, you know, relative to this maximum, and 20% in D3. And then my orange or red color here, it's picked up in detector 3 and only a little bit of it in detector 1, right? And so if we look at this and you know, look at 1% and 2%, we read this is how much of the signal, let's go back, is actually picked up in this detector from fluor 1 and how much is picked up from fluor 2. And, you know, likewise, If we look at floor 3's emission, 1% goes into detector 2, 0% or is detector 1, 0% to detector 2, and the maximum in detector 3, right? So if we pivot this or rotate this graph counterclockwise by 90 degrees, we get a similar looking view, but now we can make a matrix math problem out of it. So we are actually calculating how much fractional percentage is picked up in each secondary detector relative to the primary detector for each fluorochrome. And those are the numbers we get, right? 1.15 and 0.01 are the numbers that we get out of the spillovers. And we have to solve this system of linear equations because we're measuring only the total amount of light picked up in each detector, and we want to know how much of that is contributed from each fluorescent probe in the system. So to do that, you do some matrix math, and this is not something to be done by hand. Even a 3-color matrix is a very tedious process to do by hand, but the computer can do it quite quickly for us. And we actually measure all of the spillover coefficients. That's what we can get, and then we invert that matrix to create a compensation matrix. So the point here is not that you need to know matrix math, but that it's not as simple as just subtracting away the spillover coefficients. You're actually inverting a matrix and creating this compensation matrix, which is the inverse of the spillover matrix itself. Now, the other point I want to make is that this basic example is exactly what we do when we have full spectrum cytometry, or a very similar mathematical equation is used to unmix in full spectrum cytometry, right? So we still calculate the spillover matrix for all detectors. The difference between traditional cytometric approaches is you have space between the filter sets, and the filter sets are designed to only pick up like the peak emission of and you aren't considering the entire spectrum. And here on a spectral instrument, we have these filter sets basically side by side. There would be room for the laser lines, but you're trying to pick up as much of the spectrum as possible. And so the math becomes a little more complex, but ordinary least squares still is just basic compensation math, and we still have to solve for the same problem or get the same information, which are the spillover coefficients. That is how much fractional percentage of this peak emission gets picked up by all of the secondary detectors in the system. So when you're going to create a compensation matrix, it's really important to have good compensation controls. And this information has been around for years. Some of the key opinion leaders have talked about it on the Purdue cytometry list. And I like to share these good rules of compensation controls with everybody because they've worked well for me, and I think they'll work well for you as well. So you have to have that single stain control for every fluorescent probe. Every color used in the experiment has to have a single stain control. But additionally, you want to have the compensation controls matching the exact experimental fluorochrome. So FITC is FITC, whether it's conjugated one antibody or another. But FITC is not GFP. And if you try and compensate with a FITC antibody for your GFP mouse, it's not really appropriate. You will not get a perfect solution there, right? So you should use a GFP cell line or something of the sort to pull that emission spectra of the GFP, not replace it with something else. Tandem dyes are very lot specific. So APCH7 from lot 1 going to be a little spill over a little bit differently because of the conjugation efficiency of the tandem dye together varies from lot to lot during the manufacturing process. You go to a different lot, you could have slightly different spillover values. So you should always be lot specific, use the exact same tube of antibody with any of those tandem dyes for the comp controls as you use in the real experiment when you're staining your cells with all of the antibodies together. The second point is that background fluorescence should be the same for the positive and negative control. So the simplest way to think about this is if you're going to use cells, stain them for your comp control, and you get a positive population that you define, the unstained or negative population should be the same cell type, right? It should be the same cells. If you're using beads as comp particles, the positive beads should match the negative beads in their autofluorescence levels. And this is also important to think about when you're using monocyte markers versus lymphocyte markers. Monocytes have different autofluorescent properties. So if you use CD14, you want to move your gate up into that region of monocytes to get the CD14 expression, but your negative Control should also be unstained cells gated in the monocyte region, right? And your lymphocyte markers should be unstained cells gated in the lymphocyte region. Just know that you never want to mix and match beads with cells for positive and negative fractions when you're setting up your compensation, because that autofluorescence changes, and therefore the slope and the calculation of the spillover coefficients that we need to calculate the compensation matrix changes. So always match beads to beads, cells to cells, and within your mixture of populations in a cell sample, use the same type of cell for the positive and the negative for each control. Now, controls need to be at least as bright or brighter than any sample the compensation will be applied to. This is really important if you have dim antigens or dim controls, and then you have much more expression in your actual all stains. You can get some really wacky skewing if you don't get a control that's as bright or brighter than the sample is going to be applied to. For this, I like beads because you can always add a little more antibody to comp beads and it'll push the signal up. The population will go up, up, up. You can put it all, usually won't even saturate. You can push it off the scale. And you want to, check your controls and kind of do a couple rounds of testing with them to make sure that you can get good compensation before what I call shooting the moon, which is, sacking $10,000 worth of mice that you've been working with for 2 months and then realizing that you had poor comp controls because you didn't do some iterative testing or development of a panel, especially important with higher parameter panels and spectral cytometry panels. If you use any type of fixative or permeabilization reagents, these can change the autofluorescence and fluorescence properties of the cells and the antibodies that are attached to them and the fluorochromes that are attached to those, right? So all your probes can change slightly or even break down a little bit, especially with the tandems. If it's a really harsh detergent, you could get some breakdown. So if you're going to fix and perm your all stain, fully stained samples, you should treat your compensation controls exactly that way too, so that everything matches. You're trying to capture this emission as it is on the actual experimental cells, and you want your comp controls to match that experiment as closely as possible. Now, when you're defining initially what is positive and negative, and for each color, right? Again, we're going to define a positive population and negative population here. You're going to have to define a cleanup gate, or what we call in FlowJo a size gate. It's going to be on a scatter set of parameters in a bivariate plot, and we're going to pick either the lymphocytes or the monocytes or the granulocytes. Here is linear scaled scatter. Some people like to put scatter on a log scale. It's up to you, but you do want to define that size, that population first. And again, if my marker's on lymphocytes, I want to gate here. If it's my marker's on monocytes, I want to gate up here. And grands are going to be up here because they are going to have different autofluorescent properties. And we're going to want to match the same type of cell to positive and negative fractions here. So focus on that population with the expression of the antigen targeted by your antibody fluorochrome conjugate or probe, in this case, your fluorescent probe. Traditional compensation is all about defining the right positive and negatives. We're going to draw a gate around a subset of cells that is stained with a fluorescent probe, and a gate that is on the negatives that are not stained with a fluorescent probe. And then we're going to calculate a slope, and we're going to calculate a spillover coefficient from that removes the contribution of this di X into all secondary detectors one at a time, right? So looking at bivariate plots, it's easy to see. We've got a negative and we've got this positive that is now bleeding in the uncompensated state to detector 2. And what we're going to measure or remove is the spillover, right? We want to bring this population down so that it is on the same line or has the same MFI in detector 2, both positive and negative. And here's the compensated state, right? After applying the compensation, you do get this phenomenon of spillover spreading. And spreading is part of cytometry. It's innate to the detector system that we use and how we measure fluorescence. We're, limited by the noise in the detector that causes this spreading. And we'll talk a little bit more about spreading later on, and how we can evaluate the spread that is going to occur when we two colors together in a staining panel. So we do calculate the spillover coefficients, and then we invert those with a big matrix math problem to get a compensation matrix that removes the contribution of this fluorochrome into non-primary detectors to equalize the medians. And the result of that, depending on the combination of fluorochromes and their properties, the detector system, the cytometer that you're using, is going to result in this flaring effect or spreading of the population, which essentially reduces sensitivity in the detector that you're spilling into and compensating out of, right? So again, median of the positive and negative should be equal post-compensation in this compensation control. And that seems easy, right? We just gate positive, gate negative, and no problems. You press a button, it calculates, right? It's always not quite that simple. Difficulty becomes with compensation controls that are not ideal, right? If you have low frequency of expression, cells may not be the appropriate compensation control. If you have low antigen density expression, that is not a very bright signal, right? That might not be conducive to a good compensation control. And then we have this problem of autofluorescence, where some cells are more fluorescent in certain regions of the spectrum than others, and that can lead to incorrect compensation if you don't take that into account. So I'll give you an example here from Carlos Roca's paper on AutoSpill, where they've removed autofluorescence and improved the detection of MHC2 in microglia from mouse brains. So here in this example, I've got this rare antigen. The antibody is conjugated to FITC. Here's what you would see in FlowJo, which is a histogram. You're going to try and gate. You need to gate at least 2% of the events from the size-gated population that I'm displaying here in this plot. Unfortunately, this antigen is expressed on so few cells in this system that I can't really accurately gate for positivity. If we turn that into a bivariate plot, look at another marker where this FITC is spilling into the BB630 secondary detector, you see this skewing up as you go up in FITC signal, you get the skewing up. And I could gate it here, but what we're trying to do is define a point where we're going to draw a line through the median of the negative and the positive to define the slope and create the spillover coefficient, right? So this isn't a very good compensation control. So I'll ask the question, is it better to move the gate on this compensation control with very few cells actually expressing the antigen of interest? Is it better to just shift the gate over so you get 2%, but you're actually not gating positives? You might be gating on an edge of the negative. In this case, it's not. It's actually reducing the MFI of the median point that we're going to draw that slope through and changing the calculation. So in this case, I might suggest to use some beads, right? We can bind the antibody fluorochrome conjugate to beads, make a nice positive signal, and then we can calculate a slope through it. So there's no perfect solution sometimes when you have low antigen density or low frequency of expression antigens that you're working with. But cell comps are not always conducive or the best things to use for compensation controls because of those issues. Just to see. So we have a question here. Some of the markers are low or very variable between human donors in experiments, and in bigger panels, we need to use cells rather than compensation beads because of the brilliant dyes don't seem to work well with compensation beads. Some people are using CD4 conjugated with brilliant dyes rather than actual antibodies of interest. How much of an effect is likely to have on comps? And are there any other solutions? That's a good question, Andrea. I recognize you. If it's not a tandem, you're fine using another antibody, right? If it's FITC or PE or any monochrome like BB421, go ahead, use CD4 conjugated to that color. If it's a tandem dye, of which all the other BVs besides 421 are, PE size 7, right? You really want to use the lot-specific antibody fluorochrome that you are staining with. And so I'm not aware of that challenge with compensation beads. There should be, I mean, there are many different bead manufacturers. Maybe there is a good solution there for binding those antibodies. There are different beads, like rat hamster beads are different from the anti-mouse beads. So you got to look at where your antibody comes from and get the appropriate bead for the antibody that you're using, right? But that is a very good question. Thank you for posing it. So let's go ahead to the next slide here. AutoSpill compensation. What are we doing with AutoSpill? We're simply removing the positive and negative fraction gating, and we're now using a linear fit or robust linear regression to draw that line through the sample and then calculate the spillover coefficient. So it is the exact same math, right, to create the compensation matrix that is inverting the spillover coefficient matrix that you calculate here with all of these single colors. But it's removing the need to calculate or to define a positive and negative fraction. You still use those same single stain controls. All of the rules apply for good compensation controls. And then, no positive negative gates are needed. Simple as that, right? It seems really, really basic, but yeah, it's a Nature paper because it was an innovation that makes it a lot easier to work with lots of samples and not have to go through and check your gating positive and negative. You still need to find your upstream size gate. You got to say, I don't want to gate on everything because I've got debris, lymphocytes, monocytes, and maybe grands in there. You want to make a size gate initially and define that as the population you're going to use for the linear regression calculation, but then you don't need to, inside of that size gate, define the positive and negative gating or fractions. So robust linear regression just simply weights less the events that are further away. So you get a better regression line. It is an iterative calculation where it like fits and then it does it again and does it again and tries to converge on a solution that is optimal. In most flow plots, we're looking at log scale data, and that's why you see the skewing kind of as a hook. If you then were to transform and put your scaling into a linear scale, you would see that it's a nice straight line. And so this figure from Carlos Roca's paper is just showing that traditional compensation and AutoSpill, just the first step of linear regression that AutoSpill does, gives you a very, very similar solution, right? So it works, is the point. Now, iterative fitting can actually improve the linear regression. So AutoSpill actually compensates, it refits the data, estimates error, calculates a new fit, and repeats to convergence and does the same thing, calculating the spillover coefficients that we then use to calculate the compensation matrix. The real benefit of this method, when it was first implemented, it was the first way that we could offer an autofluorescence subtraction of the cell's autofluorescence in a system. And the autofluorescence spectrum of cells is basically treating the unstained cell sample of the exact same cell type you're interested in, right? It has to be cell type specific. And it's going to estimate the autofluorescence and use it just like it was a probe, right? But with this iterative robust linear regression, we can calculate a slope of autofluorescence in a cell sample. And this may not be a great example, but I will get to some demos that show you more. And the key point here is that you have to have the unstained sample of the same cell type that you're actually interested in to accurately calculate autofluorescence. And you have to have a detector that is open, that is no other stain is used as, that detector is not used as a primary detector for some other fluorochrome or fluorescent reagent in your system. And you have to choose a parameter which has high autofluorescence, right? I can't just pick a red detector, leave it open, and expect my lymphocytes to show autofluorescence in it. That's not where they autofluoresce. In the paper, they suggest using violets and UV channels, and that's what we see most commonly. But you need to actually run these unstained cells and look at them and say, where is the signal the highest? Where is it most autofluorescent across the spectrum? And I'm going to use that detector. So Roca says, sacrifice the highest autofluorescence detector during your panel design and use that as the autofluorescence channel. So traditionally, we have this spillover spreading algorithm that depends on the defined positive and negative populations. And so when we implemented AutoSpill compensation in FlowJo, our engineers came up with a solution to partition the data and estimate the fluorescence and actually get the standard deviations and calculate a linear fit and be able to go ahead and calculate a spillover spreading matrix. And we'll talk a little bit more about spreading matrices, and I'll refer to you two are very good resource for understanding what spillover spreading does, how it's used, and what total spreading error can be measured now as well. And they have different purposes, so we'll get there. But right now, I just want to say we do calculate a spillover spreading matrix. And for most data sets, when we're using AutoSpill, for most data sets, this AutoSpread calculation, as we're calling it, produces a spillover spreading matrix that is very close to the traditional spillover spreading algorithm originally published by Roederer's group. So why would we use this AutoSpill, AutoSpread? Well, simply less gate adjustments on the controls. You don't have to define the positive and negative. You still have to define the size gate. That's the population of events that you want to use for the calculation. But then you don't have to define the positive and negative, which makes things a lot easier. Ideally, this method should get it right because we're iteratively calculating the regression line. It's as good as it's going to get on the sample that you're in the population you've defined to calculate on. And it shouldn't require manual tuning, right? Well, we say that, the authors say that. In real life, everybody wants to tweak their matrix for one purpose or another, and we'll talk a little bit more about that as well. The big benefit is this autofluorescence removal, right? With that extra channel, you have to have a channel open that has high level of signal, or at least the most signal autofluorescence in the unstained control. And as I said in the paper, they say sacrifice the most autofluorescent channel during panel design in order to enhance resolution across all the other channels. The limitations, well, it performs linear regression in a tube with an internal positive and negative, right? So it really works best when we're using cell comps in a single tube, as opposed to mixing and matching the positive and negatives or using a universal unstained control or comp beads. It will calculate on beads, right? But it was really designed to use cell comps. Some data sets don't converge on a good solution with this iterative slope, and it depends on how messy the comp controls are. But you know, sometimes that can be compromised with low levels of fluorescence. So again, the same problem with a very low antigen density expression or a very low number of events that are positive. It could, you know, it's tough to calculate a slope from that type of data. So consider using an alternative. And of course, autofluorescence cannot be calculated on a bead. It's completely different autofluorescence levels than your cell populations. So always use cells, unstained cells of the exact same type that you're most interested in the system to remove autofluorescence. I do have a question, which is in higher dimensional panels, we've suggested to our users to match the cell type if you want to sway antibodies, noting that they are not tandems, using CD19 PE instead of a lower expression B mark, but this doesn't work with tandem dyes. So yeah, it's somebody making a comment about Andrea's original question. Thank you, Ariana. And that's exactly what I said. If it's PE, use a different marker that has a nice positive signal, no problem. PE is the same no matter where you get it. FITSI is the same no matter where you get it. But if it's any tandem dye, you have to work out a different way to get it. And that's what I revert back to. Beads usually, but in most, full spectrum, cytometrists are trying to mix and match depending on their needs. And if you have to go to beads, that's fine. You got to do what you got to do to get a good signal. It's better to have a nice positive signal, I think, than a poor one. Simple as that for calculating compensation. But it's a good point to make, right? So here's an example from the paper with AutoSpill improving the detection of MHC2 on microglia. And the take-home point here is pretty simple. With traditional compensation and no removal of the autofluorescence, you get an overestimation in the microglia populations, which is the red traced histogram here. It's higher MFI than the CD4 T cells, which don't have any MHC2 or any other knockout. So they've got knockout animals, and they're comparing it to splenocytes with lots of MHC2, right? And if you were to draw a gate based on your FMO on the splenocytes, right, and compare all of these other tissues and conditions, you might overestimate or cut into the microglia population, which really shouldn't have any MHC2 on it. But when they use unstained microglia as the autofluorescence subtraction sample in an AutoSpill compensation calculation, it more accurately removes that autofluorescence contributing to this secondary detector, and now you get no, the correct or a more appropriate resolution and a more accurate biological conclusion. And I'll show you this as one of my examples of how they set this up in the demo here. The paper is really good, though. If you're going to use this method, highly recommend to read the paper about it. Same with spillover spreading. Go read the paper. The math might not be to your liking, or we aren't mathematicians, most of us, but the take-home points in the discussion of these papers, even if they're full of math, are really good for cytometrists to understand why we are using these tools and approaching the compensation problem in this way. So I'm going to go through a couple slides, just how to use compensation in FlowJo version 10, and then I will go and jump into a live demo with a few different data sets so that we can go ahead and get some real end user interaction with the FlowJo user interface, and I can show you how to set these up appropriately. Another question: if we are following the rule of the comp control is brighter than it's applied to, do we still need to use the fix per cell as the comp control? For example, can we use arc beads to set up the comp control for a fixable dye? Thanks. Arc beads are amine reactive dye beads, and I've never seen them be perfect. I would much rather take a sample, heat kill half of it, mix it back together and make bullets, and then pull a bullet of half heat killed cells that have actual biological cells that are either dead or going to pick up the amine dye or not. You still want to always treat your comps like you treat your cells. And yes, if it's beads, in my lab, I would fix and perm the beads because I'm trying to treat the antibody fluorochrome conjugate. And if there's any breakdown of that tandem dye fluorochrome due to the harsh buffer system that I'm using, I want to recapitulate that even if it's on beads. So let me give you an example. Actually, I'm going to give you an example here. I've got a few different comps. Some of them are beads, most of them are beads, and others are cells, but some are surface antibodies that I've stained first, then fixed perm the cells. So those antibodies and their fluorochromes see the fixed perm. And then after the fixed perm, I go in with the intracellular markers. Well, my intracellular antibodies, they don't see fixed perm, right? So even though I'm using beads for almost everything here in this experiment, I'll show it to you live in a sec, I'm only fixing perming the comp controls for the antibody fluorochromes that saw fixed perm in the real processing of the all stained samples. So only the surface antibodies and their fluorochromes see the fixed perm or get treated with it. The intracellular antibodies with their fluorochromes, they never saw that reagent. And so when I process the comps, I'm actually doing it all in a plate, all at the same time, and I'm treating the comps exactly like I'm treating the controls, right? So intracellular antibodies don't see fixed perm, but comps don't get fixed perm. Just wash. Extracellular or surface antibodies, they see fixed perm. I'm going to give them fixed perm comps as well, right? Hope that makes sense. Tough question here. The question now is the focus on a panel involving both lymphocyte and myeloid cells with some sharing markers, should two unmixing compensation matrices be used when analyzing either lymph or myeloid subpopulation? In a perfect world, I would say yes. And I know that's not a fair thing to ask everybody to do, right? So what you absolutely have to do is gate the right population and get the right population for your markers, right? CD14 is on monocytes, it's not on lymphocytes. You get the monocyte region and get a signal. Should you calculate two different sets of compensations? Depends on whether your autofluorescence subtraction channel needs to be there for both populations, right? You could split it up and create two different matrices, one that is specific with an autofluorescent subtraction of monocyte fluorescence, and another that is specific with an autofluorescent subtraction of lymphocytes. In real practice, that's a lot of work. The other options, you could gate those populations, export them as individual samples, and then apply different compensation to them in FlowJo, right? That's possible as well. I don't think many people do, and in all honesty, I would ask your core director or the other people using your instrumentation in your institution that question and have a debate or a good conversation about it. I can't tell you whether yes, go for it or not. It depends on your real world use case. But it is true that when you subtract autofluorescence using one population, you're trying to look at different populations in a mixture, there's always going to be some that are not ideally compensated, a population. So it's a good question. I don't think many people do go that deep, right? Most of the time you're trying to look at myeloid lymphoid, you pick you're always going to pick the populations that you have, that you want to look at most, right? I'm interested in this question about this subset. I also want to look at these, but really I'm going to focus on this as my main core. And so that's what I'm going to compensate for and do autofluorescence subtraction for. I can still get the frequencies of other populations. Maybe they won't be ideally compensated, but is it good enough? And am I ever going to actually use that information on that other subpopulation is the other problem, right? Oftentimes we cast a really wide net, and then the story you write up in a paper is about a very small subset of the data that you've collected. So you might not go there right away unless you need to talk about that population. But it's a tough question, right? And that's the whole point. It's gotten much more complicated with the expansion of detectors, the advent of full spectrum cytometry, and the number of probes that we can use in an experiment have gone so high that we want to look at everything, but maybe it won't be perfect in that one experiment. You might have to break it down. Certainly different tissues, I might consider doing unique compensation controls because the autofluorescent properties of my splenocytes are different from microglia in the brain. And so consider that. What's the question you're asking? What is the cell type I'm most concerned with or interested in? And at least compensate for that cell type, and then worry about the other things later downstream if you're going to need to talk about them or discuss them. Okay, so when you load your data samples into FlowJo, you get a list of samples, right? And there's this square box next to all of the samples. I call it a badge. If you double click on any of those squares, it'll open up this matrix preview / edit window where you can actually select any compensation matrix. The default matrix that comes in with the samples is always called an acquisition defined matrix. And an acquisition defined matrix, I have a cat who's yelling in the back. An acquisition defined matrix is going to be gray. And when you calculate a matrix in FlowJo, you're going to get a color matrix, and you can name it something specific, and you can calculate multiple matrices from the same setup or modify that setup a little bit. And again, all you have to do is select a matrix, grab the M button, drag it onto a sample or a group, and it'll apply that matrix to that data. To preview the sample, you can pick from the drop-down list, preview a population that's already gated on the sample, so you get down to a specific subset and see the effect of the matrix on the data. Overlay uncompensated is checkboxes here. It shows in blue. The event color depend on the color of the comp, and you'll see them change if you multi-select compensations. You can actually compare them and see how they're different. By selecting two compensation controls, you'll look at the deltas and see how they're different from each other. Now, initiating compensation is a click of a button, but you do need to put your compensation controls into a compensation group. I also recommend actually size gating, at least initially, and applying that size gate to the group. Then when you click the compensation button, it's going to auto gate for positive and or negative fractions. and assign each of those samples to the channel that it's brightest in. The next step is you need to verify that all the channels are being used. In this case, I have an extra channel. It's blank. It was not stained for. With traditional compensation, I'm going to remove that parameter. Removing parameters can be done from this drop-down list. Click on any of these sample drop-downs, and at the bottom, there's a bunch of options and one of them is just remove this parameter from consideration. So I remove that parameter, and now I'm down to what I've got is just a 9-color experiment on a 16 detector instrument. Pretty simple, right? In this day and age. And we have these options now. We can either use traditional, that'll be the default radio button over here on the right. That's the spillover algorithm that we're going to use. That means we need to gate for positive and negative effectively, or we can click on AutoSpill, just a click of a button. Now we're going to remove the gating and calculate linear regression on the size gated population that you have defined here as the positive. Spectral is another checkbox where you just check the box and it's going to now use all of the detectors. You can actually limit which detectors you are considering in the spectral calculation. Under this button, all detectors, you have another list. If I want to deselect a few of them because not consider them for any reason, you can do that. And once you've checked the boxes, you can give your compensation a name. And then when everything's set up properly, you'll see a finalized message in the top right-hand corner. Clicking the View Matrix button will take you to the next step where you actually get the compensation. So it is important to always review and adjust your gates. I will give you several examples of this in the live demos. Here, I've got an amine reactive dye, stained cells. I'm using unstained cells as the negative and then stained cells as the positive. But if I left the size gate that is created automatically on the lymphocyte population, there's almost no dead cells there. So I actually have to move the gate down a little bit and forward scatter to capture the dead events that are stained with the amine reactive dye and get up to a fractional percentage above 2% to get a green light that says I'm good to go with this color, right? Let's see, we got some more questions, so I want to address them. No, same thing. I'm sorry. Yes, and Shemi, the recording will be available, and I will show that to you here in just a moment where it is. Go to FlowJo's website, search for recorded webinars, or give us an email at flowjo@bd.com. We can link them to you. It will take a couple days, probably mid next week. This will be available to review. So these columns are where you set all of the sample and then the positive and negative. So you have to choose the sample that is the single stain control for the color or parameter. Then you have to choose a positive and a negative with traditional compensation, right? That is defining the positive and the negative. You can choose to use a universal negative for all your beads or all your cells that are unstained. US means unstained here. No fixed perm means no fixed perm because this was an intracellular antigen. Yeah, and that's actually not true. I don't mean to misspeak, but it's just in this screenshot, that's what I'm using. And again, we may need to double click on any bivariate plot or monovariate plot. It'll bring up a graph window. You can then adjust the gating that you see there to accurately gate for both size and for both positive and negative fractions. Once you are ready with your setup and you've got reviewed all your positive and negatives, you got good separation between the positive and negatives here, good staining, then you can click on this View Matrix button and it will open up that preview window and show you the matrix that you've just created. If you want to, click on a color, you can change the color. You can also you can export this matrix and edit it. So here, again, I'm selecting my matrix that I want to apply and dragging this M button to a group of samples, and you will see the color of the badge next to the sample take on the color, this lattice work, be the color of the compensation that you've applied. So click on the color next to the name of the compensation that you want to use. You can change the colors so that they pop, and then you can go back and forth and see the effect of what you've done in your compensation and how it affects your data simply by applying the matrix to the samples and then viewing them in a graph window. Now, again, editing compensation can be achieved in this window by clicking this edit button, and you want to match the positive and negative fractions so that they have equal MFIs in the spillover channel, and that would be a correct compensation. The editable copy is automatically applied to all the data, and I want to warn you that if you have a 35 color experiment and 100 samples, when you change this value, it recalculates every event that this compensation is applied to for all samples. That is very memory heavy. So I actually like to click this edit button, select acquisition defined, and apply that matrix back to all my samples. I can still preview any sample, and then I can edit the copy of the matrix where it's only updating the one sample and updating the profiles here, not recalculating every event and every gate in my workspace. It's a lot faster. You'll see the change that you make, and you can evaluate it. You can apply your matrix to one sample and then evaluate that, right? In a graph window too. As soon as you make a change, it'll change the skewing of the population depending on what interaction you're editing. And we read this matrix just simply as the rose fluorochrome is bleeding into the columns detector by some amount. So just a warning, 100 samples, 30 color matrix, editing it with the matrix applied will take longer to see the updates and recalculate everything. Now, when you calculate a spectral matrix, there is no longer a one-to-one relationship between the fluorochromes and the detectors. And unfortunately, this makes it very hard to edit a matrix. And the math is correct based on the inputs. Should you edit this? I don't think it's appropriate. So what most groups are doing, manufacturers and such, they're giving an option to export the compensated data set. That is unmixed data, and you'll only get the compensated the channels that are the primary detectors for each fluorescent probe, and it won't give you every detector on the instrument. Instead, you're just going to get a matrix of zeros or no matrix at all because it's just compensated events. So there's no longer a one-to-one relationship. I can't go, this is bleeding into that, and I'm going to change that number, right? Because the actual calculation is using the entire spectrum, and there's many detectors that are secondary, and it's all converging to a good solution. So what you can do, as I said, is if I come in here with this spectral compensation matrix, I could just choose to export compensated values from FlowJo, bring that in. You won't have a comp matrix, right? But you can actually click this plus button in the bottom left-hand corner and create a matrix of zeros, just a blank identity matrix. And then you have a one-to-one relationship, right? 33 by 33 as opposed to 33 by 48. That is what Aurora data comes in with, right? And that is what you can do with spectral data from an A5 SE BD instrument. Just simply apply a matrix of zeros to the unmixed exported files or exported as compensated in FlowJo. And then you can tweak the relationships and cause the change in skewing that you really want to create or adjust with that editing of the matrix. I'm just looking at a few more. Selecting spectral the same as unmixing in Diva. The answer is yes. So spectral unmixing is what it's doing, and there are several different ways to spectrally unmix. But yes, you are still doing the same thing, selecting spectral unmixing on a A5 SE instrument running Diva 9.3. It's doing a spectral unmixing calculation. There are two ways to export data from an A5 SE. One is just as FCS, where you get this big acquisition defined matrix, that is the number of fluorescent probes by the number of detectors on the instrument, right? 33 by 48 in this case. Or you can export as unmixed FCS, and then you only get the unmixed data. So you get the compensated data, no compensation matrix in that case, but you can always click the plus button, slap on a square matrix of zeros, and then edit the individual interactions between fluorochrome to detector here. One more question. What does the spectral option do for the compensation? Why would one opt for spectral compensation? Oh, that is the million-dollar question. I don't think spectral is the solution to everything. If you are doing a simple, sometimes traditional is just easier, right? You have fewer detectors to worry about. You're only taking a sliver, so you have less error compounding into the matrix calculation and less spreading. You'll notice with spectral instruments, you get a much wider negatives, and we deal with that by compressing around zero in the bi-exponential transform function, setting a higher width basis, which compacts the data around zero, but then you see 10 to the 4 to 10 to the 4, right? And that's all negative, and you got a couple decades above that. So spectral comes with its problems. I would say if you want to resolve very fluorochromes that are very, very similar in spectra, in emission, spectral is the way to go. But if you're doing a 6-color experiment, I'd go with my 16-color Fortessa and just get the data. It's going to be great, right? And I don't have 48 detectors to worry about. So you use the instrument that you have access to, and you can use a spectral instrument in a traditional way just by only considering the detectors that are the primary for each reagent in the calculation of compensation, or you can consider everything. So there's no perfect answer to that. Again, I think you want to go to the experts at your institution or company and have that conversation. Which instrument is the best for my purpose? But you don't, just because it's the shiniest thing in the world, or everyone else is using it, doesn't mean that it's better for your application, right? So think about that when you're designing your experiment. You have an intent in mind to look at a specific set of features on the cells, and you want to use the instrument that is conducive to making really nice, pretty readouts. I've seen some data on spectral versus traditional instruments, and it's so wonky and uninterpretable on one, and it's really good on another. Like, why force it? Just use the one that's easiest to get the answer and has the cleanest data to make your biological conclusions. Great questions, guys. So saving a compensation matrix, there's a Save Matrix button. Just know that you can export it as a CSV or a FlowJo matrix file. And if you do a FlowJo matrix file, then you can import that matrix by clicking the plus button. It'll ask you want to create a new identity matrix or bring one in from a file. The matrix MTX format just has different header information that FlowJo can read, and you can transfer your matrices across different workspaces or bring them in after exporting. But best practice is always to compensate fresh with each experiment using fresh compensation controls collected at the same time as your all-stained experiment, because you're capturing the performance and setup of that instrument and the spillover that occurs. And there's drift on instruments. I don't care what your AppSci for another company says, that we can just use a bank of controls that we've already calculated. Best practice is always to create fresh controls, run them at the same time as the experiment, and then apply compensation based on those controls to that experiment. Now, spreading matrices have been introduced into FlowJo in recent versions. This is 1010. It has the most options, including spillover spreading matrices, which are independent of MFI or brightness of the controls, and mostly used to monitor instrument performance. That is the argument made by the authors of the paper. And they are calculated now, as I mentioned, with either traditional or AutoSpill matrices. Okay, so you can view these by clicking this SSM button and display or export as a CSV the spillover spreading matrix. Higher numbers mean more spreading, right? But again, this traditional spillover spreading matrix calculation is independent of the actual brightness. So it is not a good representation of the spillover that you will get in a specific panel with different antigen densities for different dyes. You can change the antigen density, that is, put that fluorochrome on a different marker that is brighter or less, and you'll still get the same number here, which means it's really about instrument performance and consistency. It's not about panel design. So what do we use for panel design now? BD came up with this total spreading matrix or total spillover measurement, right? Which when we get these numbers that are turned into a matrix, the numbers are much higher. There's their unit list as far as I know, but it doesn't matter. The bigger number, the worse the spreading is going to be between two fluorochromes, or when you double stain those populations with those dyes, you're going to get spreading that reduces the sensitivity in the other channel, right? And you want to avoid those combinations. Or at least you want to have very bright things in a channel that spills into and spreads a lot. Or else if you have a dim thing, right, it's going to overlap. Now, I don't have the slides to articulate this at all, but I do have a very good webinar from BD Biosciences, from Mirko Corselli, on total spread measurements and the difference and the application of these two spread measurements between on flow data. And it's about an hour long. You can find it at the BD Biosciences website. Need to get a link, email FlowJo@bd.com and we'll send it to you. If you just search for, spread measurements in BD Biosciences or total spreading matrix, you'll find it as one of the top hits on Google. And the total spreading matrix does take into effect or into account the MFI of the entire system. So it is panel specific, and as you get brighter, you will get more spread, or dimmer, you'll get less spread, because spreading has a relationship to how bright the dyes are. In FlowJo, though, at this point in time with 1010, it is only calculated, total spreading matrix will only be calculated when you use a traditional algorithm. So it's just a click of a button. If you want to try AutoSpill, you click the radio button for AutoSpill. If you want to try traditional, click the traditional. But if you're going to get a total spread matrix for use in iterative panel development, do the traditional method and you will get a TSM option. Otherwise, it just won't be here, right? And these spillover spreading and total spreading matrices, they're calculated from the comp controls. So that information does not come in with an acquisition defined matrix. If you click acquisition defined, you will not have these options, okay? So again, really good webinar on that topic, recent webinar. If you want access to it, just let me know. So cosine similarity is another feature that we've implemented. This is how close are the spectral profiles of different fluorochromes. And this can also be used to kind of determine which dyes you should use in combination for a high parameter panel. The closer to one, the more close those spectra are across the instrument. And so you probably want to avoid color combinations that are really close to each other. You get better resolution generally when you're working with colors or dyes, fluorescent probes that have low cosine similarity and distinctly different spectra. Having said that, many people do use spectral cytometry to differentiate between very closely related probe spectra, and you can still do it. There's no set cutoff that I'm aware of that says the big rule that if it's over 0.85, you shouldn't use them together, right? It's up to you. But this is a calculation that is kind of tedious to do if you have to export all the MFIs from your comp controls and do it in Excel. And now it's been implemented in FlowJo, and it's just a click. I mean, it's just there when you calculate the matrix. So again, this is a pairwise contra. So it's only a half matrix is needed because they're just duplicated. All these values are just mirrored over the other side. But it's a pairwise comparison between the emission spectra of each fluorochrome pair. The values will always be between 0 and 1. High cosine similarity, that is closer to 1, generally indicates higher compensation values will occur and increased spillover spreading will occur between those fluorochromes. And if you use fluorochromes that have low cosine similarity, that will generally produce better results, lower compensation values, and lower spillover spreading. So I hope that helps to point you towards some additional metrics that you can use for instrument performance over time, that's spillover spreading, for panel development, that's TSM, and for similarity between fluorochromes, that would be the cosine similarity. So searchable documentation resource at flowjo.com is available. We've got our recorded webinars here. You can get our plugins, which I didn't talk about today, but on the exchange, and then flowjo@bd.com. I keep saying that's our technical support line, and we can get you the right materials or send you to the right websites to get this information. So if I click on recorded webinars here, it's going to open up for me, right? Here's recorded webinars. If you go to the Learn tab, the Webinars tab, here are the future webinars. This is the one you're joining right now. Please do, join us for others in the future. And then down at the bottom is recorded webinars of this Learn webinars page. We've got a recorded webinars link, and it just takes a couple days, but all our recent recorded webinars are listed here. This is the website on the BD Biosciences site that is the SSM TSM discussion, and it is a very good webinar if you want to use those tools for, again, instrument monitoring, performance monitoring versus panel design with the total spreading matrix. I found it very insightful. Other resources, I'm going to show you this 33 color panel. That was the big matrix. This is actually a set panel used for immunophenotyping of T, B, and NK, as well as dendritic cells in peripheral blood. There's a tech note on it. It is a standard panel that's available through BD Biosciences. If you want more information, you can actually just search for it, or again, you can email us at flowjo.com and or flowjo@bd.com, and we will link this to you if you would like. Right? So now I'm going to jump into a demo. Don't feel bad about. Okay, I do have a question about, it's unclear to me what you were saying about having all the channels exported when you export this way. So we'll see. Somebody else, okay. If spectral unmixing doesn't look right, or is it okay to export as FCS and then redo the spectral unmixing in FlowJo? Does that give the same results as Diva spectral unmixing? The answer is yes, you can. It just takes a little while to set up in FlowJo, right? So if you do it correctly on the instrument, defining your positive and negatives and doing the spectral unmixing, that's great. You can also export as unmixed, then apply a compensation matrix of zeros in FlowJo and edit the interactions one-to-one using that matrix. So I think it'll make more sense when we get into some of these demos. I know I'm already over time, but I'm going to keep going for everybody that wants to join me to see how we actually interact with FlowJo here. So let's go ahead and fire it up. We're going to open the application. We're going to use a few different examples, and we'll talk about some of the options, right? So my simple example is just my classic 9-color experiment on a traditional cytometer. You only collect the channels that you're using. If there are additional ones, you can remove them. I go to my compensation group. It's got all my comps. Why? Because I put the word comp into the sample name. So FlowJo's looking for anything that has COMP or unstained in the sample name, and it's going to automatically put it here. At this point, I can go ahead and just press the compensation button. After selecting the group, it auto-gates size, and FlowJo tries to stick each sample where it's brightest in each brightest channel, right? So it does a good job when there's a limited amount of channels, and the region of the spectrum is very discrete that we're looking at. It's harder on a spectral instrument. You have different gains and voltages for different sets of detectors, and things will go to the wrong detector because it's brightest in that one, even though it's not what you considered or designed the primary detector for. I have a blank channel here. I can remove this channel, it's gone. I need to then go ahead and view all of my positive and negatives, and I see one at the top that is still a problem. Everything else looks good. Beads are matched to beads. Cells are matched to cells. But in this case, I don't have any dead signal. That's because my size gate is on the major population. The dead stuff is a little smaller in forward scatter. So I'm going to double click on this sample and bring it up in a graph window, move my gate a little bit lower on the scatter, and there's my dead stuff in the lower forward scatter region. And I can now work with that. How do I move this? There we go. There's all these bars from the zoom that I need to move around a little bit. So now I'm good, right? It's looking for the name of the detector in the name of the sample. So ideally, you do put the name of the parameter that is the target parameter for your comp control in the name of the sample. That's really helpful, especially when we get to 48 detectors and 33 colors, as I'll show you. But my calculation is ready and it's finalized, and I can go ahead and view the matrix. All right, and now I have a compensation matrix. It's very similar to the compensation matrix that I was, calculated on the instrument and brought in with the data. Very, very similar, right? It's the same data. Should be similar if you gated the same thing. I have some negatives. So personally, I like to run unstained cells and unstained beads, but it's only with traditional compensation. If you're using AutoSpill, it works best with internal negatives. So, that is a decision you have to make for yourself. I would still collect unstained cells and evaluate whether the internal negative is better. Here, obviously, an internal negative would give a different slope calculation because this is an amine reactive dye, and so even the live cells get coated with the reagent, and so it's actually brighter than the completely unstained sample. Shouldn't matter with the live dead, but it might matter with another sample. For all these others, you can see my unstained, internal unstained, that is the green trace here. I'm going to open this one up just as an example, is identical to, right? So here's some cells, they're unstained. The blue trace, which is my completely unstained universal negative, is exactly the same. So I love using universal negatives because they're easy when you're doing traditional cytometry. But again, if you're doing AutoSpill, you should probably have the in-tube negative. But that makes it difficult if everything is positive, right? CD3 on all the lymphocytes. Does what's left over in the negative have the same autofluorescent properties? Mostly if you correctly gated lymphocytes, but it might not be the case for monocytes. They're all going to be CD14 positive. There's nothing left for an internal negative. You can always concatenate two samples together, an unstained and a stained, and then you have it in the same tube or the same file, right? But the events that are negative came from the unstained sample, and the events that are positive were from the stained sample. So there's ways to get around the challenges, but you have to think about it, right? And think, what's the best comp control I can make for this? Because accurate, good controls lead to accurate compensation calculations. All right, so that's all I'll say about this one, because I want to get on to a more complex example. But traditional cytometry is pretty easy. You got a size gate, you got a positive and a negative gate. Use beads, match to beads, cells match to cells, and always use lot-specific tandem dyes for your comps. Now, let's just close that. I don't need to save it. Let's use a different example. So AutoSpill, right? AutoSpill, for the sake of time, I'm not going to go from scratch. I do have the demo down, but it takes a little while because these, samples are not labeled ideally. So instead, I'm going to open up kind of a pre-canned example here. And this example is the data set. It's available on Flow Repository as part of the Roca paper when they originally put that data up. And that's where I got it. And then you have to kind of figure out where the channels go. So I have a little script. I know where they're supposed to go, but that's what I'm saying. Like, if you're just labeling, I love that they put cells here, right? I like to know that it's cell sample or a bead sample. It makes it easier for me to work with. I love that the fluorochrome is in the name, but the thing they're missing is the detector name on the instrument. So let me just double click here and I'll show you what I mean. We're double clicking on this matrix setup, right? The traditional matrix, these data came in with just nothing on them. And if I calculate an AutoSpill matrix, I can do that either with or without an extra channel, considering that unstained microglia sample that is the autofluorescence of the microglia. But to actually set this up, I've got to start by initiating a size gate. So I like to gate my cells first, apply that gate to the compensation control. So you see I've got this cells gate. I've applied it to the compensation group. And then when I click the compensation button, it just gates for positive and is going to try and use unstained cells as the negative. This submenu that comes out when you calculate your first matrix, that is the setup in the last set state you set it, you left it in. So if I double click on this sub menu, said compensation in orange, that'll get me back to the setup of this calculation. And you can see I've got 1, 2, 3, 4, 5, 6 different detectors, but this is a 48 color instrument, right? Or 48 detector instrument. Do I need to do spectral unmixing? No. I could use it as a traditional instrument and only consider the signal in each discrete detector, or I can do it as unmixed, or as spectral. I can calculate using AutoSpill that removes the gates, right? So again, I've defined positive for each fraction, or I can remove it. And ultimately, the take-home point of this data set, friends, is that this V8 channel using unstained cells that are the actual microglia cells from the brain that have autofluorescence in this V8 channel. You can see this little blip or population, right? And it's going to contribute to other channels. There's the key point, is that you have this autofluorescent subset of events in this sample that actually will overestimate the amount of MHC2 if you don't account for it using autofluorescence subtraction. And so this is a great example of just not having to gate anything except the size gated cells. So I don't want the debris, I don't want the big stuff, I want the actual cells that are intact. And then we go ahead and calculate the matrix, and it's just doing an iterative regression analysis of each of these samples using one sample that's in tube negatives, and it's calculating the entire subset, right? If I do that without this autofluorescent subset, that is, I remove the V8 autofluorescent parameter and calculate the same thing, then I get two different matrices, right? And I've already done this. I've got an AutoSpill and autofluorescence, right? So I've got AutoSpill without the V8 autofluorescent channel and the unstained cells being used to subtract that out. And then I've got one where I've used that V8 autofluorescent sample. Now we go and we look at the actual overlays in a graph window here. What I'm going to do then is apply the matrix that I've created with AutoSpill, but no autofluorescence subtraction to those all stained samples. And we get this same problem, which is an overestimation. Sorry, I'm moving my lists and things here. Is an overestimation of the amount of MHC2 compared to the knockout MHC2 knockout. So these are both supposed to have no MHC2 because they're from a knockout animal. Unfortunately, the brain microglia subset has autofluorescence, which leads to this shift in the MFI of the peak of these cells. And this is due to autofluorescence, not due to the actual expression of MHC2 in this animal in microglia, right? Obviously, because it's a knockout brain. But the moment we use that autofluorescence subtraction, use violet 8 channel and unstained microglia to compensate out the autofluorescence of that subset, when we apply that matrix with autofluorescence subtraction, you can see it shifts down and overlaps. And so that's the whole take-home point of that particular figure from the paper, is that autofluorescence subtraction works, and it can give you better more biologically relevant results. So last example here, I'll go ahead and close that unless there are any questions on it. I see a lot of you still on, so thank you very much for sticking with me. Let's see if there's any other questions. No, not yet. So this is now a 33 color panel. I'm not going to save that one. I'm going to open it up. Now, why don't I start from scratch here, just to show you the challenge and why I love labeling compensation controls correctly, or rigorously. So I've got lots of controls here, right? And a few all-stained donors. I'm going to bring them all into my sample. Notice how they all the comps, all 33 compensation controls go where they need to be, which is in the comp group because it has the COMP in the name of the sample. And additionally, this user who collected these samples has annotated the actual primary detector that they would like this comp control to be associated with in the name of the sample, in addition to the actual target antigen and the color, right? So we see YG730. That's the name of the detector on this instrument. You'll see in just a minute. So what am I going to do here? Sure, I want to start again with size gating, right? So let me draw a little size gate. I don't have to be crazy with the size gate. I got plenty of cells here, right? I'm going to call it size. Doesn't really matter. I could call it cells. I could leave it lymphocytes. And then we go to the compensation group with that, right? So now I've applied that all the way. Is that appropriate for every sample? Almost. They're all cell samples, right? I'm scrolling through them here. But oh, I got some beads. All right. Well, that size gate's not going to work on the beads. So let's go over here and grab some beads, right? That's all I need to do is move it or adjust it for those samples and capture some beads. I could be more rigorous with this, redraw the size gate just for those samples or something like that, but I'm just moving them. Again, notice the beads are actually usually on these rare antigen populations that just don't have enough cells in the system that are positive to make a good compensation control, so they go to the bead system instead. But most of these are cells. Here's one more bead. And then I'll get to one or two which are not lymphocyte markers, right? And I probably have already passed, but we might see this later. This one's really wonky, and I'm just going to cheat here. Oops, by grabbing more cells. And I know this is kind of, I didn't make these samples, so I don't know. But for some reason, there's only 7 AAD signal in these cells that are really high. All right, so first thing, check the size gate. Do I have the appropriate population? If I get to something like CD14, let's see, it's in here somewhere. There it is. All right, so my CD14 gate, right? Is there going to be anything in my YG660 detector? Not much. Why? Well, it's CD14. I've gated on lymphocytes. So let's go up, move it over to an area where there's CD14, and boom, I've got my positive and my negative there, right? So just remember that it has to be specific to the cell type. Now, my next step is go ahead and view this calculation. So I've gated on size, I've adjusted size so it's appropriate for every sample. Now I'm going to go ahead and click my compensation calculation, and it goes, oh man, we got 48 detectors here. What do you want to work with? You can choose it here. You could select only the 33 detectors that you want to consider if you're doing something traditional. It's a spectral instrument. I'm doing a spectral calculation here. I'm going to choose all my detectors, and then I'm going to assign them to the correct detectors. And in reality, this is going to take just a minute, but it's got to calculate, it's got to auto gate, it's got to assign these samples to the brightest channel. And it's not going to always get it right because, again, you're now taking every bit of information from the entire spectrum, and these detectors and their filter sets are just side by side. And the gains and the PMT voltages that are set up for different detectors off of different laser lines might actually result in brighter signal off of a laser that it's not supposed to be on. And it's really weird, but that's what happens. I see it on all different spectral instruments when just looking at data. And of course, I always see the data. I don't get to make it anymore. So I'm kind of just dealing with the data as it comes in and trying to share with you my insights as to, how to make this work. One thing, as I said before, which wasn't in the ROCA example data, was the detector name or the parameter name. It's really helpful to label that in the comp control name so that you can accurately assess whether it went to the right detector. And in truth, you could just compensate with those in any detectors, and it's not going to matter because everything gets calculated to the brightest detector no matter what, even if you assign it to a different one. The 100%, the calculation that we then say, oh, this fractional percentage is the spillover into all the other detectors, it's all based off of where it's brightest. So it doesn't matter which one it is, but it's really weird as an end user if your PE size 7 is coming off of a violet, right? Or a UV channel. And you're like, what? That's supposed to be off of yellow-green. Why isn't it off of yellow-green? Well, the answer is it went to the brightest thing. As the whole sample, it goes to the brightest detector. And it may not be right when you're using full spectrum. It's a lot easier to assign those, auto-assign those in when you're using discrete slivers of the spectrum and only considering the primary detector and not everything else. So here, I'm not going to go through every step. I have this pre-canned, I'm going to get there, but I'm basically going to go through and say, did it assign the right thing and do I have good positive and negatives for every channel, right? I can do these one at a time. When I click on this list, I get all the compensation controls and the option to remove or choose the parameters to consider, right? I'm going to do a spectral calculation here. So I'm going to check this spectral box. I'm going to do it traditionally. That is, define my positive and negatives. Yeah, you would just want to hit AutoSpill and go. Might work. I still want to go through and make sure everything was size gated correctly. And if I were a betting man, I would say, that will give you better results. So I want this color to be in this detector because that's the way I've designed the panel. It makes sense to me. And the name of the comp detector is going to be the resulting parameter that I'm going to then use for downstream analysis. What I like about this drop down is I can actually type in and search through. So I'm going to go B537. There's the comp. It's already assigned correctly here, right? But I need to search through it because I have the detector name in the sample name. I can accurately just find it really easy. So again, more information in the names is really helpful when you're trying to work this out downstream and do this in FlowJo. Now, this comp control, is it even right? Let's go ahead and make this bigger. Oh, here's one where it assigned a YGPE to a blue. Silly, huh? Right? But of course, this is probably pumped up in terms of PMT voltage relative to the yellow-green. And they both have the same similar emission area, even though it's cross laser line excited. So is this correct? No, it's not. Do I have a comp control that's B576? Nope. All right, well, let's remove this detector then. Scroll down, remove this parameter, right? Goes away. Let's look at the next one, B602. What's wrong with this sample? Let's go to it. Oh, it's just not auto gating because it has such weak, oh, it's 11C. What do you know? What should I do here? Is 11C on lots of lymphocytes? No, it's on monocytes. So let's go and do 11C on the monocyte population, right? And likewise, do I have anything that's negative for 11C? Maybe a little blip. So I could probably gate here and call this. My other monitor went on with this big pink color screen detector or screensaver, lit me up. So here, like, do I want to do this? Yeah, sure. I could go BB 630 neg and call it, but that's really small, right? What would be a better thing to do? Well, I do have an autofluorescent control here. Let's see where it is. This autofluorescent control, AF, right? Already has a size gate on it for lymphocytes. Why don't I just make a monocyte size gate and use that as the negative, right? Mono neg. All right, so now I've got that. I can actually pick from the drop down for negative my unstained autofluorescent control, which is going to be assigned as an autofluorescent control in the UV446, but I've now gated for mono negatives. Boom. Now I'm using monocytes that are negative, appropriately unstained, right? And monocytes that are positive for the antibody. And now I have a good positive and negative. Is it conducive to AutoSpill with an in-tube negative? Not as much, right? So where, is it better or worse? I still like traditional compensation. Maybe I'm just stuck doing it because that's the way I learned it. But that is an appropriate setup, right? I'm using matched positive and negatives with the same autofluorescence levels. I'm just choosing from a different tube, which I'm double dipping here to use for autofluorescence and as a negative for monocytes. But that's a much more appropriate setup than if I were just to leave it gated on the lymphocytes and then tweak my gate over to, get 2%. So I hope that makes sense. All of these other ones, I've got green lights because it's finding the detector name, which I love, right? And I can go through and just make sure that it works. Is this an appropriate gate? Yeah, would work. I might move it over like that, right? Be a little more specific. Again, I'm defining the positive and negatives, and we're using medians to calculate the slope. So I want those to be most accurate I can. And the auto-gating algorithm is just a peak finding algorithm. It doesn't always do the perfect job. All right, do I have an R660? I sure don't. So I'm going to go ahead and remove it. Right, and I'm just being processual through here. APCH7, it's in the R. Do I have an R680? Nope. I'm going to remove it. At the end, I could go through and only assign things that are positive, but I find it easier to actually remove parameters if I'm not using them as primaries. At the end, you could go and just say remove unused parameters, and anything you haven't assigned a tube to be a control for, then it drops it out of consideration for the list of antigens, right? Again, we're doing antigens times all detectors because we're using spectral, right? I'm using all detectors for this calculation. But what I've found is it's easier to go one by one, evaluate, do I have a control here? Here's another one, R710. This is a BV711 fluorochrome that the author of this data and panel decided should be in the violet, but it's assigned it to a red. So I'll get there, but basically I'm going to go, do I have an R710 control? No, I don't, so I can remove it. Again, remove this parameter. And then if I go down to an R or a violet 710 detector, right, here's my V710. It didn't get assigned anything. It's got an antigen label, but I can go V710, and there's the comp control it's supposed to be appropriately in, and I then click on the label here, and it'll show me this one was used as a bead, right, but a nice control. All right, so that's basically it. You just have to be really processual because you're working with so many more detectors. And if you want them to be assigned to the exact thing that you design the panel that is the primary parameter, readout, or channel, you have to make sure that they go there. Here's the autofluorescence channel, right? So I want to make this point here. Autofluorescence is a property of the cells that are unique, right? In this case, we're looking at the size-gated cells. That's what we're on. But I need to actually define what is positive and negative, right? So I'm taking all the cells, basically everything. There's almost nothing that's negative. So what can I use as a negative? Well, in the latest version of FlowJo, we've implemented a true negative. It is at the bottom of this list for negatives, right? We got this negative column. We go to the autofluorescence channel. It's got a red light because it says, I don't know what to use as the negative. In this case, with traditional gating, we can set true 0 as the negative, right? So we have positive autofluorescent cells, true 0 is the negative. If we go to AutoSpill, we don't need it. It removes all of these gating. It's not considering it, right? And it's just going to do a regression line through that sample. And it should work fine. But if you're doing traditional gated populations with autofluorescence subtraction, the purpose of this true negative is to give you a negative subset fraction and say we would assume that we want to compensate out all of the autofluorescence. So it's at 0. Again, name of the UV515, it's there. I can pick that compensation control correctly. Then I go to it and I look, why isn't it giving me a green light? That's because it didn't auto-gate it properly. So I need to move this over. With traditional, I need to define a negative. So I'm going to draw my own negative and call it BU or UV 515 neg. Right? And then once I have that negative defined manually, I can go to that channel. Here's the drop-down slot. And in the middle section here are all the gates on that sample that you've assigned. And I can choose the negative slot, and then I get a green light. Right? One at a time. That's all you can do with it. Here I've got another YG that's going to a UV channel. Automatically, it's not appropriate. Is there a UV 540? No, there's not. So I can remove that. And obviously this takes time. So that's why I'm like, I wouldn't redo it unless you have to. Like if you can get it right on the instrument first, use the acquisition defined matrix. I don't see any problem with that. Recalculating everything in FlowJo will take setup time. Now, once it's set up, you can calculate multiple matrices just by clicking these buttons, right? You can uncheck spectral and do a traditional and only consider 33 detectors, or you can check spectral and consider all 48 and do the spectral and mixing math. You can check autofluorescence instead of traditional, or remove the gating that you've performed and set up, and do linear regression, and you can make a couple different matrices. Now I'm going to close this. I'll save it in case anybody asks a question and we want to come back to it, right? So let's go 33C and call it good on my desktop. But I do have a pre-canned 33 color one, right? So I've done the exact same thing here. I've created several compensation matrices. I can double click on the submenu from the compensation group here, and now you can see my setup. I'll try and spread it out as much as I can. And again, I saved it in a state to make the specific point about autofluorescence that you do need to assign a negative if you're doing traditional, and that true zero option is at the bottom of the list for your negative dropdown. So just assign it as a true zero. Now I've got a point from which we can calculate that line through the medians and create a slope and a spillover coefficient. Love those green lights though, right? Everything's been assigned here. I can double check anything just by clicking on the color. I mentioned 7AAD as being a wonky one. I prefer amine reactive dyes personally for live dead reagents, but and again, if you're going to do a live dead, might as well heat kill half the cells, mix them back together with the live cells, then you got a nice positive population that's going to be stained with your live dead reagent instead of very few cells in a healthy sample, right? So as I can again check spectral. It's going to do a calculation in the background, takes a little more time for the math, but it'll say finalize, and then I can view the matrix. So I can call this, spectral, traditional. Can't type, I guess. Hit my cap locks. But you get the point. Name it something specific, and you can view the matrix. And then once you've viewed the matrix, you can go ahead and apply it to your data. So I've done this a couple times on this data set, unmixed using traditional gating with a true zero for the autofluorescence. I can unmix using AutoSpill and linear regression to calculate the slopes and spillover. I can unmix again, and again, and assign different colors and apply these to the data for my all stains, right? So right now it's on the gray acquisition defined matrix, which is this really big spectral matrix from the instrument. Nothing wrong with this matrix. It was a good matrix, right? Did I need to do this again? Not really. But as an example, I'm using it to say, label your comps. When you have 48 detectors, it really helps to put the detector name in the comp control. That is, where's the primary you want to go to? You got to be processual, right? Start with all detectors, remove the ones that you aren't using as primaries one by one, and verify the positive and negative fractions for each channel, right? And you can just do that by clicking, right? Everything looks pretty good. Again, this one, I hadn't set it up right. I would be better. It always helps to look again. This is where CD11C is, right? And look, now it wants to auto gate 0.82%, but it needs a 2%, so that's not good. I just showed this to you, so I won't show it again, but use the unstained, gated in the monocyte region, assign that as the negative. Now you have a good slope calculation. That would probably be more conducive to AutoSpill. And once you have this compensation matrix created, right? Again, it's 33 by 48 here. I can't edit this. I mean, it'll allow you to edit the matrix. It'll make a copy and you can tweak the numbers, but you don't know how you're really affecting the overall state of the cells. I know you want to just make it straight, not skewed, or maybe bring what's negative up so you don't have a negative MFI. But the math is correct underlying it. So I go back to, would I trust the math or do I trust my eye? We're so used to doing that with lower parameter traditional matrices that have a one-to-one correlation that we want to do it here. But if you wanted to do that, I'm going to show you this to you now. You might as well go to your Allstain and export the darn thing, compensate it, and then tweak it. So what I'll do is I'll apply my true AutoSpill detector matrix. It's not perfect. And then I'll take my sample. I'm just going to do 100,000 K events. And I can export that, right? Export or concatenate populations. We're going to export all events to the desktop. And I'm going to choose all compensated parameters, right? Or you can go custom set of parameters and choose which ones you want, right? I want my scatters. I want all my comps, whatever you want, right? So all compensated, I'm going to export it. I'm going to bring this into a new workspace to just keep it clean. And that workspace is right here. Right, so I have no compensation anymore because these should have all comp parameters. Yep, FJ comp, that denotes that we exported the compensated data. Now, if we go to this matrix preview in this new workspace off of the sample, you notice there's no matrices, right? So what do we do to tweak? We're going to add a new identity matrix using this plus button. And we're going to select all of the compensated channels. And it's going to create a matrix of zeros. And now I can start tweaking things. So I actually am getting into demo. I didn't practice this earlier, so I don't know exactly what I'm going to use, but I can take any color, slap a gate on here, look at that population and say, well, what's something that's going to bleed into something else, right? Let's look at BB755, right? Versus, well, I don't know. Let's look at CD4 versus CD8. Right, CD4 versus CD8. That looks pretty nice, right? Where is my CD4? Right here. Where's my CD8? More parameters, more challenges, right? And it's like you got to be familiar with your panels to really be quick. So that's the R780 and B537. So many channels to work with. R780. Yes. So I'm going here into R780. Now what happens when I do, 25? I need to apply this matrix, right? First to see the effect of it. What happens if I put 50 in here? Oh, see, I don't even know. This is called demo-itis, where I haven't played with this and given you a perfect example. But I promise it works, right? And this is what Cytec teaches you to do on an Aurora, right? They give you a matrix of zeros and say, edit the interactions here. I just haven't set it up properly to give you a good example. But you can add or, put in 25 and then add a negative to it, right? Is that appropriate? Oh, you can see the effect here. So I love that. Forget about the graph window, right? So what did I just do? I made a tweak and I made things skew in a way that was not appropriate, right? Just to show that it works. I would rather stay with the same sample, right? And this negative 25, right, is going to affect this guy. So let's just put a 0 here. And the reason why everything is completely overlaid is because, this matrix of zeros, every point is exactly the same. The moment I do something to this matrix, you'll see a difference. What it did was remove more signal here into the B660 channel from the contribution of B75, or it added signal, so it removed more, compensated out more signal, and now my gray dots, that is this, the compensated, this compensation matrix applied events are different from the original, right? Which were the correctly compensated events. And so it is possible to edit a matrix, but it is best done when there is a basically one-to-one relationship, and that is achieved by exporting the compensated data, putting on a matrix of zeros, and then tweaking the interactions to cause skewing or remove skewing, as the case may be, for your particular panel and color combination. Do we have any other questions here? I know I've blown through my 1 hour, turned it into a 45-minute demo. Classic Tim Crawford move. If some of you are core directors that used to have me in the lab giving presentations, I hope this was helpful. Can we use these data to practice compensation with this nice 33 fluorochrome panel? I will have to ask that. I don't know if the actual data is available for me to share with you. I will ask the scientific affairs team if that is okay to give these samples out. It is a really pretty data set. They did publish this as a standard panel that you can just use off the shelf. And it works really well. The only deficiency I see is the 7AAD comp is a little challenging because it just doesn't have a lot of dead cells in the healthy cell area. Another question, but I will look up for you. And why don't you send me an email at flowjo@bd.com and ask me that question, and then I will talk to the powers that own this data, and I will find out if it's acceptable to share it with you. And if so, I'll share you OneDrive link and you can download it. Another question, can I just import unmixed files and add an identity matrix? 100%, you can. And that's the method that we promote off of an A5 SE instrument or any other spectral instrument, is export as unmixed and then slap on a matrix of zeros, which again is just creating an identity matrix using only the compensated parameters. But if you export as unmixed from a Symphony A5 SE, you'll only get these channels, right? They will say SW unmix instead of comp. That's the way they decided to write it. And then you can apply this matrix of zeros and edit. Yes, and so Diva does have two options if you have a spectral enabled Symphony, and using Diva 9.3 is the most recent version, I believe. If you right click on any selection of samples that you've acquired, there should be two options there. One is export as FCS, and in that case, you get the raw data with a spectral unmixing matrix that you've calculated on the instrument. That is this acquisition defined matrix, which is the number of fluorochromes or probes by the number of detectors, right? A big rectangular matrix. Or if you right click and say export as unmixed FCS, then you get, it's like exporting as compensated parameters only in FlowJo. You get only the compensated data, and then you will have a 33 color experiment. It drops all of the unused secondary detectors that were not assigned as primary detectors for each fluorochrome. And it's a simpler way to do it, right? Then you slap on a matrix of zeros in FlowJo, and you can still edit those one-to-one interactions. All right. Well, thank you guys for your time and attention today. Do appreciate you hanging around with me for 2 hours. If you have other questions, reach out to our tech support team, and we'll do our best to support you and your research goals. So until next time, have a great day. Good luck with your research projects, and thanks for your time. My pleasure, Andrea. Thank you, guys.